And today I am going to talk about chromatin remodeler. So as you know that Benoit has started with this chromatin remodeling BAP60C and has gone long way. Today I'll be talking about another chromatin remodeler of the same family, and this is called Brahma, and what it does during differentiation. So my slides are not taken. So this is a slide that I borrowed from Elphez, that when he said that he wanted to study differentiation and development, but still stuck with ESLs. But we have done a little bit more work in the term of differentiation. So as you know that when a sperm fertilizes a egg, it makes a zygote, and that continuously differentiates and develops to form terminal cell types. As cells differentiate, they increasingly lose potential to go to other lineages, and they restrict themselves to a particular multipotent and then finally unipotent cell types. This is beautifully explained in this Waddington diagram, where on the top of the landscape is an undifferentiated cell, and as it differentiates, it takes these canals and forms and takes a particular canal, encountering bifurcations, and forms terminal cell types like blood or heart or neuron. And if you don't get anything from this today's talk, I want to tell you that this chromatin remodeler protein Brahma that forms these ridges so that it uh, prevents cells from going away from the cardiac canal to other canals. So that's why we say that it safeguards cardiac sulfate canalization during mesoderm differentiation. Uh, Brahma is part of a Swiss large multi-subunit complex, along with another protein called BRG1, which I won't be touching much today, but we have done much work. And here in Bruno Lab, Jeff, and previously June, and Heiko. So this is the catalytic subunits, BRG1 or BRM, with some core subunits and some variant subunits. And these variant subunits, they associate with different types of cell types. Mm, Many of these complex members are involved during mouse cardiogenesis at different stages, and mutations in these members are also implicated in congenital heart disease. Using a cardiac differentiation system, what we found that during ES cells to cardiomyocyte differentiation, the complex composition of this complex or the BRG1 complex changes. And then we isolated complexes containing a cardiac enriched subunit BAP60C, BAP170. BAP60C is also another cardiac enriched subunit, but for this work, we isolated complexes using BAP170, and then found that during CP to CM transition, the SMARCA2 or Brahma protein gets increasingly associated with BAF170, while SMARCA4 or BRG1 gets decreasingly associated. So that asked, then we asked that what's the role of BRM? Up until that point, BRM's role in cardiogenesis is not much known. So we started with the simple experiments asking that if BRM is okay, so I like to give what's its relations with BRG1. You can see that this is a uh, graph from Jeff Armstead's cell paper, where I'm showing the expression of BRG1 in red and BRM in blue. You can see that BRG1 is still highly expressed, where BRM just starts to express from day 4 in very low epichems, and goes and so gradually increases. So we first asked that what's the role of BRM in cardiac differentiation, and for that we did CRISPR knockout of BRM and did a cardiac differentiation. What we found that in wild type cells, you can see that the cells are very nicely beating, but these videos are playing, but you can't probably see that any signs of beating. And these are two different clones of BRM knockout. We performed RNA-seq to understand that what are the gene that are getting up or down regulated, and we performed RNA-seq at 3 different stages during cardiac differentiation: at day 4 mesoderm stage, and CP stage, and CM stage. And mesoderm stage, we do not see any changes in gene expression, but at CP and CM stages, mostly they change. And what I can say you that in BRM KO cells, mostly neural genes are upregulated, and at that point we say that okay, why is it happening? And cardiac genes are downregulated. So we didn't understand much from those gene expression data sets, and we wanted to know very clearly that if neural gene upregulation is a true thing or not. And for that, we did a single cell RNA-seq in wild type and VRM cells at day 10 of cardiomyocyte differentiation. What I'm showing here in red is the wild type cells, and the blue is the KO cells. And if you go through the markers here, which I put together, that these KO clusters are mostly enriched for retinal neurons and different other kind of neural cell types, as well as osteoblast and fibroblast, whereas wild type cells are mostly giving us cardiac myocytes or immature cardiomyocytes or epicardial cells or some cardiac myocyte related things. We did staining of beta tubulin 3, which is a pan neuronal marker, and we found that wild type cells they make cardiac myocytes, KO cells they do not, but wild type cells do not form beta tubulin 3 or top 3 forming neuronal markers, but in absence of BRM they do, and here you can see this neural outgrowths. So at this point, we are clear that, okay, they are forming two different, there is a lineage bifurcation happening in absence of BRM. And to understand the mechanism, we asked that when this lineage bifurcation happens, what are the changes in the chromatin and where does in the genome bind BRM, and what factors mediate the activity, and can BRM loss be compensated by any other factors? So for that, to ask the first question that when this happens, we did again cardiac differentiation, and this time we did single cell RNA-seq at 4 different stages: at day 0 and mesoderm stage, and cardiac precursor stage, and cardiomyocyte stage. At day 0, we did not see much of a difference between wild type and KO cells, and that's been confirmed by immunofluorescence of Yamanaka factor markers, and it doesn't show difference at the day 0 stage. However, at day 4 and day 6 and day 10 stage, we do see differences. Day 0 stage, day 4 stage, which is supposed to be mesoderm for the wild type cells, the cells are clustering it together, but you can see slight changes in the cluster pattern. They're not completely overlapping. But by day 6, which is the cardiac precursor stage, you can see that wild type cells and KO cells are completely different with some mixing in between. Same thing happens at day 10 stage also. So we know that, okay, they are completely diverging after day 4 or somewhere around day 4 or mesoderm stage. Marker analysis shows that MESP1 is expressing both in wild type KO cells, both in wild type and KO cells. KO cells little bit less, which is less than 1.5 fold or less than 2 fold. And then SMART-CD3, which is a cardiac precursor marker, is expressed only in the wild types and cardiac troponin T as well. On the other hand, SOX2, IRX3, CRABBP2, all these neural markers are getting expressed and the KO cells, showing very clear-cut lineage diversifications. And so to summarize that, we found that cell lineage diverges at an early stage, and VRM is required for mesoderm to commit cardiac lineage. And VRM prevents neural lineage commitment. And in absence of VRM, there is a neural commitment happens during differentiations. We studied chromatin structure of this during this cardiac differentiation at using ATAC-seq. I'm not spending time on explaining ATAC-seq how it works, but we did ATAC-seq at 5 different stages of cardiac differentiation: day 0, day 2, 4, and 6, and 10. Day 0 and day 2 stage, we do not find any significant changes between wild type and KO cells. So this is out of question. But from day 4 onwards, we do see. But you can see that day 4 onwards, there is slight changes in chromatin accessibility, and this chromatin accessibility pattern near cardiac genes, they remain open in wild type cells but closed in KO cells. Same thing happens with 2 more other cardiac genes and globally when we put it in heat maps. However, if we go to neural cells, these chromatins are open from the beginning, and then as we progress on the differentiation to day 6 and day 10, you can see that in wild type cells, this chromatin closes, but in wild type KO cells, they do not close. And for comparison, I'm putting either day 10 cardiomyocyte enhancers or neural progenitor enhancers, and this is near a neural progenitor enhancer. We did the same thing in globally and showing here in heat maps. what we can see that in wild type cells, mostly the chromatin is open near cardiovascular and embryo development, but in day 6 and day 10, chromatin is open for cardiac muscle tissue or cardiac developmental genes and closed in brain developmental genes. And thus, we conclude that cell lineage diverges at an early stage. BRM keeps chromatin open near developmental transcription factors and cardiac genes, and BRM keeps chromatin closed near neural genes. So, since we did not see any gene expression difference at day 0 and no ataxic changes at day 0 and day 4, we concentrated mostly on the day 4 stage, where we don't see, by bulk RNA-seq, we don't see any gene expression changes, and we see minor gene expression changes by single cell RNA-seq. So this is I'm showing ataxic peaks upon ataxic regions and the motifs that are enriched. And as you can see that at day 4 stage, GATA4 and MEP2C, MEP2, these are some of the motifs, T-box motifs. These are open, and in KO they are not open anymore. When we go to day 6, same GATA4, MEP2, GATA4 and hand, these are motifs are open, and in day 10, again MEP2, GATA4. But at the KO cells, these oct motif protein, oct motifs are getting accessible. Oct2, oct8 here, REST and PAC7 and again CTCF. So that's interesting for us, and this gives some insights that what BRM might be doing. And we did S3K27 acetylation ChIP-seq at day 4, and we found again that the sites or S3K27 sites that are reduced in KO are mostly cardiac sites or MEP2 GATA4, MEP2 GATA4 kind of stuff. But the sites that gained in KO, again OCT4 and the BRN and OCT6 are again OCT motifs and OTX motifs. So we, by Western blot, we saw that POW3F1 is OCT6. This is one of the gene we chosen with a reduced cutoff from our RNA-seq data. That is repressed in day 4, but not repressed anymore in the KO cells. Same thing at day 6 also. And when we do knockout of OCT6 by siRNA in BRM KO cells, usually form beta tubulin 3 containing neural or neuronal progenitor or showing this neurite outgrowth, and they are very much reduced in absence of or in OCT6 knockdown. We did BRM chip to find out where it binds, and when we took the chip data and find out where it binds, what motifs are enriched at the BRM binding sites, we found consistently REST motifs both in day 4, day 6, and day 10 stages. REST, which is not expressed earlier, it's expressed only at day 10, and in KO, it's not expressed, that expression is gone. And here we are doing REST knockdown in wild type cells and KO cells. In KO cells, BRM KO cells, REST knockdown does not do much. But REST is a neuronal inhibitor, specific neuronal inhibitor in non-neuronal cells. And what we can see here, no siRNA and control, there is no beta tubulin 3 positive neural outgrowths, but in wild type cells, in absence of REST or in knockdown of REST, this neurogenesis, which is repressed, now coming back. So maybe this OCT6 and OCT6 and REST are two factors that mediating BRM activity in controlling cell fate. So there are two papers that we came across, which is striking, and which is POW3F1, which is OCT6. It promotes neural fate commitment via activation of neural lineage and inhibition of external signaling pathways, and mostly this is BMP signaling pathway. That's what the paper says. Another paper that says BMP-induced REST regulates establishment of maintenance of astrocyte identity, and here astrocytes means they are meaning non-neuronal identity, and this is also true in other cases. So commonly this points out that if BMP4 might be one of the candidate, and in our differentiation system, we are also using BMP4 at that stage to induce these cells towards mesoderm lineage by using VEGF activin and BMP4. And data I have not shown, we also figured out using diagram strengths that BRM activity is mostly essential from day 0 to day 4, and it's dispensable from day 4 to day 6 onwards. So we really concentrated at this stage. and then asked that can BMP4 be rescue BMP4 rescue for loss of BRM. And that's what we did here. So what we did, we took increasing amount of BMP4 and treated with the wild type cells, and I'm showing the cardiac troponin T staining here at day 10. And so increasing amount of BMP4, we see that the cardiac troponin T staining gradually decreases with increasing amount of BMP4. But in KO cells, which are not showing any cardiac troponin T, now they show robust cardiac troponin T containing cardiomyocytes. We did confirm with ATAC-seq that this global chromatin up to certain extent it gets rescued. Also, many factors that we checked by Western blot. But I'm going to show you here today that by single cell RNA-seq, we found out that we did single cell RNA-seq at Mesoderm cardiac precursor and cardiac myocyte stage, and we can see how the rescue happens in globally. So here I'm showing that in wild type cells with normal concentration of BMP4, in this you map the day 4 cells lie here, day 6 cells lie somewhere here, and day 10 cells here forming cardiomyocytes. If we have BRM KO cells at that normal BMP4 concentration, the wild type cells, they overlap with the day 4 cells. The KO cells at day 4, they overlap each other. At day 6, they are completely separate, and day 10, they are also separate from any other things. If we increase BMP4 in wild type cells to 4x concentration, while they are here, they differ from day 4, but day 6 is somehow intermingled with the other with the low BMP4 day 6, and then they form clusters here that mostly give rise to blood and hematopoietic progenitors. But KO cells at 4x BMP4 concentration, they also overlap with their counterparts for the wild type, and at day 6 they cluster along with wild type cells with low BMP4 concentration, and at day 10 they also cluster with wild type cells at low BMP4 concentration. So this shows, at least in single cell RNA-seq wise, that we can rescue these BRM KO cells by using high amount of BMP4 induction. And that we did a mathematical modeling, and we can see that how these cells travel from day 0 to day 10 as time progresses. And we were using GATA4 activity as a proxy for cardiac lineage commitment, and FGF8 activity as a proxy for neural lineage commitment. And you can see that from day 0, the cells hovers around and then comes back with high FGF4 activity, and then comes right there with very low FGF8 activity and very high GATA4 activity, indicating there is a cardiac commitment of these cells gives to cardiomyocytes. KO cells, on the other hand, they hover around there with high FGF8 activity and low GATA4 activity. They come somewhere there and then again go back and stay with a high amount of FGF8 activity and a very low amount of GATA4 activity. However, in BMP4 with high BMP4 conditions, the KO cells, they follow a near similar path like the wild type cells in forming cardiomyocytes, and go to a high GATA4 activity and a low FGF8 activity. So with that, I want to summarize that BRM acts only during cardiac differentiation. BRM represses neurogenesis by modulating both OCT6 and REST expression, and BMP4 could rescue BRM phenotype. So if I take you back to the Waddington landscape, what happens in wild type cells? Cells differentiate and take this particular canal to form cardiomyocytes, and we may say cardiac canal. In BRM cells, BRM absent cells or BRM knockout cells, these cells travels to some distance in that canal, and we think it's up to Mesoderm, because till that point gene expression is not really we cannot really see any gene expression changes, and then after that it suddenly makes this crossover of the ridge and form go to a canal that's mostly neural and form neural progenitors and neural cell types. However, this can be rescued, and we don't know when this rescue happens and how far these cells goes in the cardiac canal versus going to the neural and coming back. But this can be rescued with using a high amount of BMP4. that I would like to acknowledge Benoit for supporting me all these years, and all the members of the Bruno Lab. And this is the old picture, and you guys, we have seen everybody else in the newest pictures. And then help from Nevan Krogan's lab, Ruth, and Leo's lab in some mathematical modeling, and all the course in the in the Gladstone Institutes, as well as funding. And I will be happy to take questions. Thank you.